Thanks very much. So after the last thing I expected to be doing was addressing you all about this when we stood up last week, because this was a developing situation. But a couple of people asked me to say things. So I'm going to give two talks actually now, about half an hour each. And people do feel at liberty to go in and out. I think there's people joining online as well. The first one is to really talk about the meningitis situation with vaccination. And the second one will be about the current Kent outbreak and where we are. And I've just actually come out of a meeting with JCVI, which will be responding to a letter from the Health Secretary about a number of these things. I'm not going to say anything about that because I don't comment on things that I have personal views, but what I'm going to provide you is all information that's in the public domain, all of this. And give you some background to some of this behind it. Just a brief tribute to the people who've worked with me since 1988 on this subject, when I went to NIBSC in response to another meningococcal emergency, and questions being asked of House of Parliament. Is the National Institute of Biological Association, Biological Standard Control as it was, which was part of the Health Service, wanted to recruit a scientist to work on meningococcal disease, and I made the very unusual decision to leave Cambridge, where I'd been at the in the biochemistry department, to go and work on this problem, and that's why I'm standing here today. And this is our group when we recently moved in the Down School, and of course we're recently moving back. A couple of people I'm going to highlight. Keith has been with me since 1998. I think, and Fran a little bit longer than that. Charlene, I'll be talking about her work. A number of other people. Jeremy Carr's done a lot of work, and Odil's also another PhD student from Australia, and Odil's another person who's worked in this area with me. This was a slide I was preparing for a talk I was going to give, and I hope some of the people who are going to come to that talk, to the genomics on the genomic medicine MSc, and unfortunately I couldn't do that because I was at another emergency JCVI meeting. And I was really pointing this out for two things. When I started this review had just been written by this chap called Heikki Peltola, who's a very distinguished, good chap, Finnish paediatrician. Meningococcal disease still with us. Meningococcal disease, interestingly, emerged in 1805. 1806. We hadn't really seen it before then. And this chap, Gaspar Vissier, described an outbreak of meningococcal disease, what we think is meningococcal disease from its description. None before there, which is interesting because it's very distinctive. However, I was going to illustrate the case that it's still in the news. You may have seen this case. Our daughter died from meningitis after starting uni. This, I thought, was a current piece of news last week. I never gave that talk, and I haven't presented that slide until today. But we're coming back to that later through here. Very quickly, just to review, as I say, this is all public online. Most important thing that I'm saying in this slide, meningitis is a syndrome. It's when the bacteria invade the meninges of the, which is the membranes around the brain. Many organisms can do that to cause meningitis. Bacterial meningitis is particularly serious, and meningococcal meningitis, hence the name meningococcal, is especially serious because it develops extremely quickly, and you can go from having a perfectly healthy individual to somebody in a medical emergency or dead within a matter of hours. This makes the management extremely difficult, and particularly in outbreaks, and particularly the one we've just been talking about. It's severe without antibiotics. It's very frequently fatal, and people have severe sequelae. However, if you catch it early and treat it, people recover almost instantly and with full recovery, but it's all about early treatment. And so high index of suspicion of cases, but the disease is very non-specific. This country is good at a number of things, incredibly good at. One is counting cases of disease, and this is half the history of meningococcal disease, the whole of the 20th century. The other thing I'll come on to next is vaccination. We're really good at both those things. Thank goodness. Probably a number of people in this room are only here because of that. Dear chap, colleague of mine, no longer with, Dennis Jones, published a review on this, but you can see that meningococcal disease is an epidemic disease. We just had this data presented, actually, to the JCVI. Mostly what was called Serogroup A. Then it changed from Serogroup A to Serogroup B, and then I came in here at the start of this interesting period of increased disease. It's an epidemic disease. You can say that these cases are caused by social deprivation. This was a global pandemic, but what was going on here? We have a better idea now. Largely, I've spent my career working on this, and as you can see, it's gone up and it's gone down again, and I'll give you a little bit more information on that. Another peculiarity about meningococcal disease and the organism, and you're going to come across a couple of them, is that it has at least four distinct epidemiologies. It occurs an endemic disease, low sporadic cases. It causes a hyperendemic disease. That's why I'm here. There was a hyperendemic outbreak in Stroud in Gloucestershire, which caused questions in the House of Parliament, which caused NIBSC to want to recruit a new scientist to work on meningococcal disease. It causes disease outbreaks, well, we've just heard about that. And it also has this epidemic pandemic disease capacity. So it causes 4 different epidemiologies. Which can be very confusing because obviously, if you get a single case, you don't know if it's just a random case, a bit of a or a hyperendemic. And that's where molecular epidemiology, what I've spent most of the last 40 years working on, has been very important. Now, this is one of my favorite slides because this tells us that we frequently forget what we've already learned. This chap, J.A. Glover, Captain James Allison Glover, Army Medical Corps, he saw an outbreak of meningococcal disease in the Guards Caterham barracks in Kent. It's called Depot X or Depot X because it was in the First World War. But the important point here you'll notice is here the case is in black, but he was wise enough, using technology that he had at the time, to do a carriage study. And he found that carriage was normal. It peaked, and when you got a peak, you got an increase in disease. Now, the other thing he showed here was that the peak occurred when the temperature went down, and look, January, February, March, that was when he's got the cases, February, March, and the temperature was low, and the ration strength was high. So in other words, this is high crowding, and these are young soldiers all mixing together. So he then realized that this was all about social distancing. Or he called it spacing out, that if you could do spacing out, nothing new under the sun. 100 years later, we were still worrying about this with COVID, but there we are here. But I found this picture of him. He lived actually, was still alive the year I was born. Yes, I am that old. But he, I think, made an enormous contribution. Now, one of the things that I took with me and why I came here was the importance, what I talked to the undergraduates, of the natural history of infectious disease and understanding not just that it's causing the disease, and this is what J.A. Glover realized, but what its natural life cycle is. And that's crucial. And we often forget it because we concentrate on the patient. So I like to use the word accidental pathogen. It's not an opportunistic pathogen in my view because there's no opportunity for the bacterium. If a bacterium is acquired by a baby, it has quite a high likelihood of causing disease for interesting reasons. It has a small likelihood of causing disease in an adult, but both of those are dead ends. This is no good for the bacterium. It has not evolved to cause disease because invasive disease, no onward transmission. Onward transmission is all about living at the back of your throat. All of us, I suspect we're in the presence of, not so many because our carriage rates are low, but I suspect there's a few meningococci in the room. And there might even be ones very closely related to the one that we're seeing in Kent. But we all experience these episodes of carriage, but mostly they are not harmful. And that's normal like. So Caroline Trotter, good chum of mine, we've done a lot of work with here. And again, this is all public domain. So how do we get rid of the meningococcus? Well, obviously, the obvious thing is you can treat people with disease. That is an emergency last-minute thing for the reasons I just said. The disease progresses very quickly. It's difficult to spot. So you have to get those antibiotics in fast. I think one of the reasons why there's only been two deaths rather than out of the cases is that as soon as there were those cases where there was a high index of suspicion and people were getting treated very quickly. So obviously that's what you can do. But obviously the other thing you can do, and you will have heard about this, you can treat carriage. So you can give people doses of antibiotics and this is very efficient, but it's short term. It stops the transmission. And that's why you will have heard in the public health response, we'll talk about later, lots of people giving vaccines. And so after those military outbreaks, which have been legion, the military have these problems all over because they have military recruit banks where they bring young people together. They're all in enclosed space. When there was the development of sulfonamide drugs in the Second World War, the US Army introduced the approach of giving all the recruits sulfonamides when they arrived. Now, this is great because this is a short-term effect. They clear the carriage. There's no transmission of meningococci. You don't get outbreaks of disease. Okay, no prizes for guessing what happened. Oh, hang on. The Vietnam War, suddenly the US Army got outbreaks of meningococcal disease. This was really bad news for the US Army for all sorts of reasons. One is the Vietnam War's bad news. The other is recruiting soldiers to go there is bad news. The other is having cases of disease, and a lot of them, as you can see, is really bad news. And what had happened, it started off at Serogroup B. The colours are going to be the same throughout. And it developed to a Serogroup C. And interestingly, we now know it was the same clone. It just switched. It switched from one serogroup to another. But importantly, it was sulfonamide resistant. So the drugs didn't work, so you had the case of disease. So all these soldiers had had prophylactic antibiotics. They hadn't worked because the bug had become resistant. And now virtually all meningococci are sulfonamide resistant. Can't use them. So what they then did, the other way you can use is you can use a vaccine. And a lot of people have been trying to make vaccines for time, and everybody thinks about vaccines as doing this. I get my shot, I won't get sick, okay? Now, that is how most vaccines have been developed, but it's not how most vaccines work well. Most vaccines that work well by doing this. They stop transmission. Trouble is, people don't appreciate this. We still have COVID because the COVID vaccines don't do that. That's why we still got it. So diphtheria vaccines work so well because they do stop transmission. Virtually all the good vaccines we have that work really well work on, but they were all developed to do direct protection. So what was able to do is the US Army, people like Gottschalk and Artenstein did some work, and they grew up meningococci of different serogroups, and the serogroups were a capsular polysaccharide, which exists around the outside of the bacterium. They purified the polysaccharide, they added it to aluminium hydroxide, they injected it, and this made a vaccine. And guess what? It worked. They introduced first a poly-C polysaccharide vaccine, then an AC, and then an ACYW vaccine. And these vaccines were good at stopping disease, as you can see, which was great. Very simple vaccines. Grow the bags up, purify using cetrimide, the polysaccharide, add it to aluminium hydroxide, introduce it, problem solved. However, it wasn't problem really solved, because bacterial polysaccharides, bacteria have evolved polysaccharide coats because they're not good immunogens. What a surprise. So what that means is that these vaccines produced by what called plain polysaccharides, just a vaccine with aluminium hydroxide, they don't really work in infants at all. They only give short-term protection, which is fine if you want to prevent military recruits, because you give them the vaccine when they arrive, and that's fine, but it doesn't stop transmission. So they're no good for population immunity, because you can't give them to infants and they don't give long immunity. All the good childhood vaccines, you can have 1 or 2 doses in childhood. It gives you protection, usually like measles, MMR vaccine against transmission. Hey, good thing. Doesn't work here. Now, in the 1990s, a number of people, and actually I still think this is the Nobel Prize that should have been awarded and it didn't, a number of scientists followed on from Gottschalk's work. This is Harry Jennings and John Robbins, and they realized that you could do this immunological magic. Polysaccharides don't work because although they cross-react against B cell receptors, and you will get a plasma cell, that depletes the memory cell pool and you get no class switching. So basically poor, short-lived immune response from a plain polysaccharide. Worse than that, you keep immunized people, you lose your pool of B cells. Now, the astonishing thing is, this is immunological magic. If you take something that's a T-cell antigen, and the diphtheria and tetanus toxin, for example, and you chemically conjugate it to the polysaccharide, then it creates this immunology. It creates something that has the property of recognizing the polysaccharide, but then it recruits T-cell help and you get a proper immune response. You get class switching, you get memory cells, you get a wonder. And this is one of the few examples where the vaccine and the immunization is better than the natural disease at producing immunity. Usually our vaccines aren't quite as good as the disease at it, but this is actually better, much better. Now, this look familiar? BBC News, March 1999, meningitis 1999, the panic. We have been here before. You saw that when I started work in 1988, there was a rise in disease. That was Serogroup B hyperendemic disease in Gloucestershire. There was a wonderful headline I haven't showed here, epidemic shock for royals, because Charles and Di were living near to where this epidemic was happening with the infant Wills and Harry. Of course, the epidemic was in a housing society. There wasn't really any likelihood that the royals were going to have a problem. But it makes the news, and there's But here, this you'll notice, is William Hague, our news chancellor, old school, saw an outbreak. When he was Secretary of State for Wales, I was involved in trying to unravel a university outbreak in Wales. This is a school outbreak. As you all know, these are highly impactful on the public mind. You have young kids, young adults, school, university, get ill and they're killed. And so, you know, this really is a bad situation. Now, this chap, I was just on the call with Mary, actually, but this chap, David Salisbury, who was head of immunisation, we used to have a head of immunisation, we don't, I think, anymore, in the DHS, he realised that there was an opportunity to use these conjugate vaccines that had just been developed. So within a year, he went to the manufacturers and said, if you make me a Men-C conjugate vaccine, we will use it. So within a year, we made this vaccine. You can see the actual the start of the program was in late 1999. So we'd immunised everybody under 20. Everybody's always talking about the COVID thing. We'd done it before. This country is good at delivering vaccines, and I was around doing carriage studies when that was introduced. Everyone under the age of 20 for the schools program was immunised against meningococcal disease within a year, and it was against the Serogroup C strain, which was causing the outbreaks. Now, if we look at Serogroup C disease, what happened in the people who were immunised is that you see that there was an almost immediate effect. But not only that, in the unimmunised people over 20, the disease also went down. Now, that is a signature, as I always tell my undergraduates, rapid declining degree, going down in unvaccinated populations of a herd immunity effect. Turned out it was a very good thing. I was very concerned about what would happen to the evolution of the bug. And I said, we need to do carriage studies to find out what's going on in the transmission system. Wasn't a popular thing, actually. The Department of Health weren't interested, but fortunately the Wellcome Trust paid for it. More on that in a moment. But it was just as well, because if you immunise people, this is data from Ray Borrow's group, immunise people within a year of immunisation, if they've had a 2, 3, 4 month schedule or one dose after at these ages, you can see they've all got good immunity. If, however, more than a year afterwards, the young children have completely lost their immunity. So in other words, the conjugate vaccines are great, but they still don't really work really optimally in infants, only on the 2, 3, 4 months. However, because we did a catch-up campaign to immunise all the kids up to 20, which we only did because there was school and university outbreaks, not because we wanted a herd immunity effect, it created a herd immunity effect and that protected everyone. Otherwise, this would have been an immunisation catastrophe, but it wasn't. It was a success. And this is where we came in a bit. We thought there should be some carriage studies. You can see this particular clonal complex of Meningococcus CC11 associated with Serogroup C. Most of the disease in the BCYW was a major cause of the disease in 1999. Very highly virulent, 28 times, well, you know, 2 orders of magnitude more likely to cause disease than some organisms. But this was the carriage of that strain, which we were able to monitor using genetic techniques we'd just developed here to show that the Group C and the Group C expressing ST11 strains went right down. So this was the perfect vaccine because it introduced herd immunity and it stopped the spread of the bug. And the disease usually went right away. This was a tremendous vaccination success. And what we learned from this is direct protection, we sort of knew that would work good. Direct in protecting infants, not as good as we hoped, but importantly, it had this important herd immunity effect. So that was a tremendous advantage. Now, this was used by the by the Gates Foundation and WHO. The biggest manifestations of meningococcal disease, as I mentioned earlier, are in Africa, in what's called the meningitis belt. And it's associated with the harmattan, which is a cold, dry, very dusty, you can't see your hand in front of your face, season. And in those seasons, you can get these periodically big Serogroup A, a different serogroup, meningococci. So the Bill and Melinda Gates Foundation looked at the UK experience with Serogroup C, said, we can do something here. So they said, we'll make a meningococcal A vaccine for Africa, conjugate polysaccharide vaccine, the same type. Fortunately, we were able to persuade everybody that we needed to do carriage. So instead of infants, they gave it to everybody aged 1 to 29-year-olds because we didn't know what the carriage system was. We've done a lot of work on this, but I can't talk about it today. But the PSA-TT, polysaccharide antigen A, tetanus toxoid, produced a great vaccine and it was introduced and rolled out, and that has eliminated meningococcal disease in Africa. Serogroup A hasn't eliminated other serogroups, but that's another story for another day. Meantime, unfortunately, this happened, as you will see in other news. So some of you may remember in 2015, there began to be serious numbers, or 2012, or serious numbers of cases of what was called MenW. Now, this is what I had feared would happen in 1999, but didn't, which is the serogroup, the ST11 epidemic clone, which interestingly was the one that caused the disease in the US Army outbreaks. It was the one that caused the C outbreak, would change its capsule. And unfortunately, it did. And it had this unknown, this unusual, and of course, we're not immunizing against the W because we were only immunizing against C. Now, fortunately, though, myself and Ray Borrow were sitting at the committee that meets on these things, and I've been sitting on for since '96, actually. We saw this rise early on, and I had some money at the end of my welcome grant to do a big analysis of all the CC11 Ws at the start, before the epidemic had really taken off. And what that analysis showed was that this was a strain called the hard strain that we'd seen around and been knocking around, causing disease in Africa, been introduced in the UK, hadn't caused much problems. But this is a whole genome analysis. These isolates are essentially indistinguishable, but at the whole genome level, you can say they're distinct. But interestingly, they were very similar to strains causing big epidemics in South America. So that information went through to JCVI, the committee I was just at this morning, and they decided to change the immunization program to go from the C to the ACYW. Now, the great thing they could do then, of course, is we knew, because we'd done a lot of studies on teenage carriage of meningococci, that most of the carriage is going on in teenagers. So if you immunize the teenagers, if you use a conjugate vaccine, you get a herd immunity effect. So you know that you're going to have a very big effect. So then we rushed out in 2015 a policy to change from the Men-C to the Men-ACYW to cover all of the vaccines. And that was introduced in 2015. So the MEMCC, the single dose, was produced by this 4-valent. And this was to combat this variant that we'd identified by our genomic analyses. It's the first time genomic analysis was used in this way. You don't hear about it very much because it worked. And this is how well it worked. This was the hard strain coming in, and it went away. And maybe that was going to happen. But as you can see, what then happened with the South America outbreak was on a really steep upward curve. Now, we introduced the vaccine here. We can't know precisely what had happened, but the vaccine was introduced 2015, 16. I suspect if it hadn't, we would have had a lot more cases. And indeed, that's what Caroline Trotter and a number of her colleagues analysed. They analysed a number of different scenarios of what would happen with W. And now this is the important thing. I highlighted this today because of this important point. Between 205 and 1,200 Men-W cases were prevented. And we don't know exactly, but my suspicion it's the higher number, by indirect protection, and 25 by direct protection. That's the estimate. This shows you how important it is to interrupt the transmission system. Now, so that's all very well. Why have we got meningococcal disease at all? Well, the reason we got meningococcal disease at all is that there are 6 serogroups that cause disease: A, C, Y, W, and X. A, C, B, Y, W, and X, and B is not covered by these vaccines. Why is that? Well, it's because the B antigen is extremely similar to human antigens. So there's been a fear of producing a B vaccine, so we've never done it. So people have tried, but there've never been a lot, and big companies haven't tried it. So the concentration of vaccines has been on what are called outer membrane vesicle vaccines. Now, that's fine. Basically, this is an outer membrane vesicle vaccine. This is actually a gel I think I ran myself for one when I was at NIBSC. They're blebs that are produced by meningococci. They contain outer membrane proteins, and you detergent extract them. But the problem is that these antigens are very diverse. So instead of having only 6 types, you've got hundreds of types. And I've spent much of my life sequencing these typing strains for this. A lot of people think sequencing and typing is boring, but it's absolutely crucial if you want to understand the biology of what's going on. So these B vaccines tended to be very specific. So the search was carried out by 2 companies, GSK and Pfizer, one using a reverse vaccinology approach, this was the Slavo GSK Sienna approach, and the other using conventional vaccinology in Pearl River, which was Wyeth, is now Pfizer. And interestingly, they both came up to essentially the same vaccine by both approaches, which is a new candidate vaccine called factor H binding protein. And Chris, who's in the audience here, has done a tremendous amount of work on this. The interesting thing about that is that I remember talking to Reno 30 odd years ago. He said, I'm going to find a conserved Reno Apollio. This vaccine is one that this is still a highly variable antigen, unfortunately. There is no magic antigen that is not variable. However, the Bexsero, which was introduced recently. So these are the two vaccines. Bexsero, which we've been using in the UK, reverse vaccinology, licensed 2013, introduced 2015 into babies. It's got 4 components. I'll talk about those in a moment. Trumember, which we don't use at the UK, Pfizer, is a conventional vaccinology, licensed 2017, and it's introduced in the UK, not introduced, but it has 2 variants of this protein. So this is the Bexsero's 4-component meningococcal vaccine. It has an outer membrane vehicle, which is something I've done a lot of work with, PORA, the molecule I started working on years ago in this, and 3 other vaccines components. This was work done by a dear friend who is sadly no longer with us, Ulrich Pörgl, led this work, showing that this vaccine would have, the 4-component vaccine would have 4 3-2-1 antigens matching against a panel of European strains. And on the basis of this assessment of coverage, the vaccine was introduced. As you can see, it's not 100% at all. So, Bexsero was introduced in the vaccine, very, very good uptake, significantly reduced the disease, and we think it's got an efficacy of up to about 60%. Interestingly, it also works against some of those Serogroup W strains as well, because it's not a B vaccine, it's a B substitute vaccine. So that means it doesn't cover all Bs, but it also means it's effective at some organisms that have other serogroups like the Ws as well. These are the 4 components, FHB. Now, I've spent a lot of my time doing typing. I started off with this, and we realized we could turn this into a typing scheme, which we called BAST. And then it was developed by Karina Breheny in my group when she was here. And then Charlene Rodriguez, who was on the call this morning and who's also playing a big role in this outbreak, developed this Mendivar, a disease-associated meningococcal status. And this shows that if the vaccine is well covered, you don't get. So unvaccinated individuals, you get people with disease that match the vaccine, partially vaccinated too, but if you're fully vaccinated, you only get organisms that are not in the vaccine. So vaccine does work, but it's not perfect. Now, the next question you've got to say is, does it affect carriage? Now, another really good chum of mine, Helen Marshall, has done an incredible job studying this. And she showed that in the carriage of disease associated in vaccinated and control groups is essentially the same. So this vaccine protects against disease, but it does not protect against carriage. And we replicated that. This is Jeremy Carr's work. Both for the only people who've done it in B FHP, which is Trumember or Bexsero, there is no significant difference between the controls of the carriage rate. So these vaccines do not affect carriage. So bottom line here is protein vesicle vaccines, they might be called MEN vaccines, they're not the same. They're made completely differently, have a different influence. They provide good protection in children and adults, as long as you've given them at least 2 doses, but they do not prevent transmission. So the indication of the use is quite different. And that, of course, makes it a very different. Remember how I told you how, so that makes the calculations very different about whether you use the vaccine or not. And in a decreasing rate of disease, there was this thought, or the JCVI recommended that though they could just about make the numbers work for babies to have Bexsero, it didn't work for the older age groups because the disease rates are so low. So just a quick summary of what happened. We had these hyperendemics I talked about. Then we have this epidemic CC11, which is the blue one. Then we had a 41-44 complex, and more of that later, spreading from that year. Introduction of the MCC vaccine. We eliminated C disease from the UK. Serogroup W, Harge outbreak came and went. So we thought, phew, Maiden was wrong. There is no vaccine switch. Sadly, the vaccine switch came a bit later, and we got the Serogroup W strains coming through. But then we introduced the conjugate ACY and MENS B in infants. And that's where we got to. Suddenly, here we are, meningococcal C still with us, and suddenly we've got a Men-B outbreak. We didn't know a week ago when I stood up for you exactly what the strain was. We now know a lot more about it, and some work I'm doing with Chris and Charlene and others, and Keith, who's in the audience, of course, as well. And so I'm now going to do a little bit of diving into that. That's then. So there we are. Teenagers, young adults are not getting the B vaccine because the disease has become very rare, as you saw the epidemic curve going down. We haven't had that many outbreaks, and we know that it's not going to be effective in herd immunity. So that was where we got to. So now we've got this outbreak. What are the four? I'm going to go through four critical questions. So what happened? I'm going to talk about what the outbreak is and what's happened so far. What's being done, the public health response. You may see a lot of stuff in the literature and talking heads on news. I've tried to avoid doing this too much because the last thing you need is uninformed comment in the media in the middle of an outbreak. I hope you'll come to the view that the public health response has been well managed. Please don't listen to the people saying that this is a public health failure, but that's for you to make your mind up when you've seen the data. Now, a much more difficult question, why did this occur? What do we know about the factors that lead to outbreaks and why this has happened? And the even more difficult question, which we will never know the answers to properly, what will happen next? Again, it's very important that those of us who are academics don't overstate what we know. We have to be very clear what we don't know. I can make judgments on the basis of my 40 years experience of working this, but that does not mean it's true. You know, what will be true is what we find out. And so we have to be really careful about this, and providing advice to politicians in that situation is really tough. So just to say, it's not like this caught anybody by surprise. I mean, there are elements of it surprising, but I was involved in redrafting these, updated 2025. I mean, there is an extensive manual, if you want to look it up, of how you deal with these outbreaks based on evidence. And of course, the evidence is what we've had so far. What we've had recently is a little bit more surprising, and that stretches it. But in fact, I think although it is surprising and it's out of the range we'd normally expect, it's still within the bounds of what we understand of meningococcal disease. Remember, I told you earlier, you can get everything from a single case through to tens or hundreds of thousands of cases. So now, when this outbreak first came out, I was very worried it was going to be a BCC11, because if that comes along, and we don't have the vaccines against it, that could be an issue, because this was an explosive outbreak, which is very much like the outbreaks I was talking to you before, caused by CC11C and W. And is this going to be a similar thing, but now a B? So what happened? So the first thing, the demographic, teenage and young adults. This is a typical at-risk group. There's lots of transmission goes on in teenage and young adults. I've measured it, and we know sort of a lot about why. We also know that it's very rapid onset. It develops in individuals in a matter of hours. That's also typical. We also know that social venue and behavior is important. When we did our early carriage studies in 1999 to 2001, when the C was introduced, on either side of the W and Bexia introductions in 2014, 15, and 2018, we know that what spread meningococci in teenagers is kissing, going to pubs and clubs, and smoking. So in other words, social factors. And we know that that is exactly what. So this is often institutions with high transmission, military recruit camps, universities, it's associated with disease. Now, we also know that IMD outbreaks, as I said, occur at very different scales. However, for a UK institutional outbreak, this is big, much bigger than we are used to. We also know that outbreaks are caused by certain hyperinvasive meningococci. It's not just the capsules. I said there's 6 capsules and there's 12 in all, but there's only 6 that are associated with disease. But it's not just the capsule, it's the genetic constitution, the family of the meningococcus that's important, particularly at driving outbreaks. And as you all know, club chemistry, and I think it's now pretty clear, I don't think, that virtually all of the cases in this outbreak are associated directly or indirectly with this particular social venue. So that's an interesting case. This has all been established in the since I last spoke to you in the last week, all of this work. So let's just have a look at what my colleagues in the Health Security Agency is having to be dealt with. Now, remember that 16th of March, my birthday, was a Monday. So that was a week ago yesterday. 4 cases had developed over the weekend. As you know, these cases are sporadic, so there's a very low index of transmission. The 17th of March, it'd gone up to 9 cases. Now, you've got to see that a developing situation in a pandemic, I always say this, it's the same with, with a military situation. One thing that you know that's true about the information that you're getting real time in an epidemic, or indeed in a war, is that it's not true. Real-time information is always going to not be very accurate, particularly in the press. This is correct now. So you see, the HSA, they start talking about confirmed cases. That means there's a laboratory confirmation. Confirmed Men B cases, i.e. that's belongs to serogroup, probable cases, and confirmed and probable cases. And you can see how it developed over time. 17th of March, 9 confirmed and 20 probable. 18th of March, 50. The pace at which this is happening and the rate at which my colleagues in the HSA have to work is extraordinary. Very few of us could cope with this. This isn't just sort of, oh, well, I need to do this, or I need to do that. This is urgent. There's all sorts of stuff. I have spent the last 20 years training people how to deal with these epidemics. I never dreamt that I'd need to be actually in the thick of one of this scale, because it's tough. Day by day, every day something different. The good news is the number of total confirmed cases hasn't gone up. In fact, it went down a bit, and so it looks like this is a typical institutional outbreak. But of course, the big question is this a new strain that's going to spread and cause lots of problems elsewhere? So what was the public health response? Again, I think this was made at pace, in accordance with the guidelines, even though it was out of the normal range, and people made what I think were sensible decisions. 15th of March, antibiotics for some students with advice to staff, students and families. So everybody who was associated, this is 15th of March, of the first 4 cases, they started giving out antibiotics. Then they had to work out, I mean, it doesn't have a label on it, a case saying what it is and where it came from. You've got to work out, you have to do the shoe leather epidemiology at speed, at scale. They worked out that everybody so far had been at club chemistry, CC, between the 5th and 7th of March. This matches exactly with the incubation process you'd expect. 17th of March is only 1 day later. Last time I spoke to you, confirmed in case of the B as a causative organ and targeted vaccination program commenced. Epidemiology considers confidential thing. So they'd already got up to the vaccination stage. Of course, you've got to get vaccine, you've got to get people. This is not trivial. 18th of March, GPs nationally advised to offer antibiotics to people who've been at club chemistry and University of Kent students. Further vaccination up to 5,000 students. And 2 doses will be required for that, at least a month apart. Next day, antibiotics in Kent national GPs continued. 4 schools identified and involved. This is because the school kids went to club chemistry. They were all year 6, and we know that these groups do that kind of thing. 20th of March, we released the first genome. Now, this was done by the HSA colleagues at the Meningococcal Reference Unit, but it was released through our database, PubMLST, which Keith and I have been running since 1998. We started the. We have a huge database of information about this organism. And by that time, they'd already done 2,360 vaccinations, and 9,000 people have been given an administrative vaccine. This is all happening within a week. 21st, continual vaccination, now up to 4,500 and 10,000 doses of antibiotics. And as of yesterday, continuing refinement of cases, as you've seen, continued vaccines and antibiotics. So that's all a very, very rapid response. It's very difficult to imagine how anybody could have responded faster. And almost certainly that rapid rollout and effective targeted rollout of antibiotics have very much reduced the chances of there being because you give people these particular antibiotics, it clears carriage. Now, what is this meningococcus? Well, this was a slide I made a few years ago for a talk. And this is what I thought the dangers were. This wasn't on here. This is a global collection of meningococci, all genome sequenced, all studied, all annotated, every single locus in our database. And this is a CC11 group that was being caused a lot of disease, usually Serogroup C, some of them W. Interestingly, also causing urephritis, a new kind of gonorrhea, actually, probably. 4821, a new clone that Ming Yan, who came here a little while ago to visit me, and the group identified and defined fluoroquinolone resistance in China. We did this with a fantastic undergraduate Anna Prasmo, working with the Polish reference lab, did some work characterizing this new strain, and there's another new strain here from Africa from Meningococcal group. Those were what were the threats. However, CC4144 complex is an ongoing issue. Now, what we're able to do, what's the Kent outbreak strain? This is a page from PubMST of the isolate information. Now, this is great. We've sequenced this. You won't be able to see this here, but this is a single contig because the HSA have been able to do a hybrid assembly of nanopore and Illumina, and it's got 2,249 genes that we can characterize in minutes because that's how we developed this, 2.2 kilomega base pair. This is a very good quality genome. It has this strain designation, typing methods that we've been developing for years. But interestingly, this strain designation is like a number of other meningococci that have caused disease in the UK and abroad. So we don't know what's special about it at this level of strain designation. However, we do have the whole genome. So what's the whole genome tell us? Well, the nice thing about the whole genome, high quality genome, it's very, very highly characterized. And we Cassia Parfit has just developed what we call a link code system. And the link code system tells you at different levels how related organisms are. So the clonal complex level is up here. And then the sequence type level that I've talked about before is here. But with genomes, we can go right down these. Don't worry about these numbers. They're meant to be machine readable. But the important point you can see here is that this strain is unique until you get to this level of 67 locus differences in the core genome, which is about 1,300 of the 2,000 odd genes. So in other words, this is a unique strain and nestled within 40 strains. How does it fit with everything else? Now, this is actually good news because it's a 41-44 complex. It's not the ST11 complex or the CC11 complex that I was worried about. But this is behaving a bit like a clonal complex 11 strain. Really weird. This is very new data, but it's in the public domain. So this is a tool that Keith, when did you develop this, Keith? This is now we're using it as well. So what this does is it visualizes this by going down different levels. And what I've started off is at the level of the clonal complex, 8,700 isolates in the big circle. And then you go down in continuous concentric circles. So you can see the ST41 complex is divided into a number of groups. And then within a group, there's a number of groups. And then when you get down to this lin code, it's equivalent to within the ST485, it's 600127, it's this group. And that's where we are. We can look at that in a bit more detail, and this, you'll be pleased to know, Keith, this was presented at the JCVI this morning. I don't think I'm giving away any secrets, and there's a technical document that's going to be published probably during this talk from the HSA with this, using this analysis tool, the tool that Keith just showed. And this is the level at 600127.4, and the event is this definable as this, and there it is all on its own. So there are other groups within here which have relation, but this at the moment is unique. So we've only got one isolate from the outbreak yet. There will be others coming, but as far as we can see, it sits within this overall family, but it's a new unique isolate. We can then very rapidly use this thing called microreact. Anybody wants to look at it, it's on here. This was developed by David Anandsen. This shows that these 40 strains we got that are closely related, or 33, these are the public ones, go back to 2000, mostly in the UK. Spain is a bit misleading. It's actually Tenerife, which is politically Spain, but geographically down here. But he may well be a UK tourist, of course. Portugal, France, and then these other UK isolates. And this is our outbreak isolate. You can see it's on a rather long branch, and there's some closely related isolates next to it. If we compare it with the strain that caused that big outbreak in New Zealand, which is also an ST4441, you can see it's quite different. But these are the three most closely related ones. Now, what we can do, and it literally takes minutes to do this, is we can say, let's select these three strains and see how closely related. And a tool that Keith developed in response during when we were teaching these types of approaches. What year was that, Keith, you developed it? It was a long time ago. Genome comparator in the early 2000s. Pardon? It was 2009. You can compare every single gene in all of the organisms. And that's already been isolated. That just happens, and it happens in minutes. And this is all the different isolates. So the Kent isolates' two nearest neighbors, there are 97.42% of the genes in those isolates are absolutely identical, not a single change. And there's a number of variant loci, and they're a mixture of new allelic variants, previously seen variants, and interesting recombination events. And the really interesting one here that we've been looking at with Chris Tang, who's in the audience, is this. These pill genes encode the pili. The pili are important because they are involved in interaction with the host, in invasion, adhesion, and in interacting with complement. And interestingly, it's got one PILX gene that's different from the others. So this is still work in progress. Don't hold me to this. But we know this has got a new POR-B gene, another gene I've spent many years working on, a new PILX gene, but interestingly, the new variant comes from this clonal complex 11. PIL-G these genes are involved in glycosylation of the pili. This gene is involved in phase variation, and these genes are involved in iron metabolism. All of these genes are genes that we know about as being involved in host pathogen interactions one way or another. And in fact, we published on many of them over the years. And this just shows this PILX gene is very similar, but it is different. And it's quite likely that these tiny, tiny differences are making all the difference between whether you get an outbreak or not. And I'll come on to that in a moment. Now, Mary Ramsey, who I mentioned earlier, reminded me, of course, that I'd done this work with Vincent Janssen and Nico Stollenberg in the early days of the Medawar building back in 2004. I've been really interested in this concept of why a meningococcus causes disease, sometimes and not others, for a long time. Now, what they did, and Nico is a statistical physics, so please don't ask me about the maths, or to quote Douglas Adams, don't ask me how it works or I'll start to whimper. But he did a lot of statistical physics in this analysis with Vincent. I had a chat with Vincent about this yesterday. And taking our assumptions about meningococci, that meningococci are accidental pathogens, diseases, they get these hyperinvasive genotypes, but if they're too aggressive, they won't spread. So these are on the margin. I use the example on an interview, it's walking a tightrope. It wants to be aggressive enough to defeat your immune system to colonize, but not so aggressive that it goes over the edge and causes disease. But what their analysis showed, that if you get marginally invasive variants, could cause outbreaks of varying scale, but they will be very unpredictable, but both unpredictable in terms of when they occur and how big they are. If your meningococci don't have any diversity, you get this sort of boring pattern of randomness. And even if you put in a yearly cycle of going up and down, which happens, it's still boring. But if you have a heterogeneous meningococcal population, you get these. Now, the interesting point about this is that Vincent and Nico made predictions on this, that this is a catastrophic-like process. So this is a highly this is not your sort of random event. And what they showed there is random events, you would find the variance to the mean of clustering of case sizes would be unity. And what we found in this paper, which not really many took so much notice of it, in 2004, but never mind, as you can see, the variance to the mean is way outside what you'd expect. So this is consistent with the model that I've just showed you. So we put this in, and I've reminded me of this. I'm going to read it out so distributions such as this one that obey power laws have been associated with events such as forest fires and earthquakes. Such distributions have over-exponential tails and are perceived as being disastrous, in that most frequently only small realizations are observed, but occasionally large realizations occur. In the case of meningococcal disease, small realizations would be equivalent to sporadic cases with large disease outbreaks renting with large events. This gives me case to think that we have some grounds for some optimism that this disease outbreak won't get bigger. Now, a comment was made in one of the technical groups on, we've not seen anything like this before. Well, actually, this looks remarkably like the outbreak in Kent in the Guards Barracks in 1917. Almost exactly the same time of year, and almost exactly the same number of cases, and almost exactly the same duration. Of course, risk factors are different, the bugs are different, the population is different, but this really shows that, thing. But of course, not necessarily common. It's catastrophic and difficult to predict. And I just wanted to put in this slide some work we did with Ulrich and Heike Klaus and Matthias Frosch years ago, looking at carriage by institution. Kindergartens have very low carriage of meningococci, primary school more, secondary more, older children more, and military recruitments very high levels of carriage. So it's all being driven by transmission. So here we are. That's the end of where we've got to at the moment. We now have this question. Obviously, this is an absolute tragedy to those poor individuals who died and the ones who've been sick. And that amplifies our attention on this. But we've got to remember, is this a very rare event, or is it going to be something that becomes more common? At the moment, we don't know. I, as I said, am cautiously optimistic that this is going to be a rare event, a bit like a firecracker going off and causing this sporadic. The worry, of course, what everybody's worried about is if this is a new strain that's going to get into transmission systems and spread. I think there are good reasons to suggest that that won't be the case, but you know, we can never be absolutely sure. Anyway, so that's where we are at the moment. That's the end of the story so far. It will continue to develop, but let's hope that we aren't going to see many more cases. There may be some with people who spread out, but hopefully mostly they're going to be left to that conventional, that one event. So thank you very much for coming and listening.